Hello and welcome to my Bioinformatics tutorial series. In today's tutorial, I'll be showing how to use Phylip or PHYlogeny Inference Package to construct or build phylogenetic trees. So the first thing to do before making a phylogenetic tree is of course having a multiple sequence alignment. And a multiple sequence or a good multiple sequence alignment will give us for sure an accurate phylogenetic tree. So if you want more information about bioinformatics in general, or phylogeny or phylogenetic trees, multiple sequence tutorial about how to use the cluster W software to make multiple sequence alignments, you can check my blog at bioinformatics-made-easy.blogspot.com in the phylogeny category. In this blog you'll find also a post called how to prepare your sequences for phylogenetic tree, because you have to prepare your sequences to do a multiple sequence alignment and then construct a phylogenetic tree. And in this post I will show you what to do and what to avoid, etc. So the main mistake that bioinformaticians do is that they think the tree produced by the cluster W, for example on the EBI web server, is a phylogenetic tree. Well, I have to tell you that you can only reproduce a guide tree with cluster W, but this tree is only a guide tree that cluster W uses to assemble the multiple sequence alignment. So don't count at it, because it's not a phylogenetic tree. Okay, and of course it will give you a false results. So let's begin with a short introduction. In phylogenetic analysis, we have three methods in building phylogenetic trees. The first one is the distance method, and the second one is the parsimony methods, and the third one is the likelihood methods. Specialists in the field of bioinformatics or evolutionary bioinformatics prefer the third one, which is the likelihood methods, because these methods are the most accurate one. But the main problem, or the only problem with them, is that they run very slow, and of course that's not a problem if you're seeking for accuracy. So Phylip is or a Phylogeny Interference Package is considered as one of the most accurate packages in calculating trees. So it's a must-have if you are a bioinformatician, and because also most specialists prefer Phylip for building the phylogenetic trees. So to access the Phylogeny Interference Package or Phylip, we go to Google as usual, and we type Phylip and we hit enter. Okay, Phylogeny Interference Package here is at the second hit, which is hosted on the bioweb.pasturepod.fr, but we have to click on the second one, Phylogeny, because it's the English version. The first one is the French version, so we click on Phylogeny. Okay, this is our packages. We have a lot of choices here. We have, for example, the parsimony method programs. These are the parsimony method programs. We have the computation of distance matrix method programs, maximum likelihood method programs, manipulation and visualization of phylogenetic trees, and other programs. So, for example, in the parsimony method programs, we have Protdist, which is for protein sequences. But this program works better with closely related sequences. That's why we're going to use the Protdist, which is a method that uses a substitution matrix to measure the distance between your aligned sequences. So it says here that it's for aligned protein sequences. I will click Protdist, and we'll wait a little for the program to load. Okay, this is our program. Here you can paste your multiple sequence alignment produced by, for example, class W. And if you are just testing this program, I advise you to go to this web address, which have a test alignment at tcoffee.org. If you click this link, you'll have here already multiple sequence alignment that you can use to test the software. But if you have your own multiple sequence alignment, you can use it. Okay, we copy it and we paste it here. Okay, in the bootstrap options, you may be asking what's bootstrap. Well, I'll tell you, just because your computer can produce a tree doesn't necessarily mean that the tree is accurate or correct. This is where bootstrapping comes in handy, because tree bootstrapping is a way to check whether your tree is biologically meaningful by assessing its robustness. So, here in perform a bootstrap before analysis, we can check this to perform a bootstrap before analysis, of course. And here we choose bootstrap. In the random number seed, and it says here that it must be odd. We write 3, for example, and the seed controls the generation of random numbers during the bootstrap, because it generates random numbers during the bootstrap. Here at how many replicates, we have to write at least 100, because the number of replicates is the number of bootstrap cycles that you do normally, of course. And normally this number should be at least 100. So in this tutorial, I'll be writing just 2, because we're just testing this software. But if you have a multiple sequence alignment and you want a robust or accurate phylogenetic tree, you have to write at least 100. Okay, click on run here, and wait for the result page to appear. So this is our first result page. This is the first section of my video tutorial. You have to check the second video to complete this tutorial. Okay, I'll see you in the next video.